Thanks so much for joining me for this webinar on CRISPR-Cas. Today's focus will be on teaching about CRISPR-Cas using gene therapy as an example. My name is Allison, and I work on the curriculum team at miniPCR Bio. miniPCR's overarching goal is to make biotechnology more accessible. And we make biotech equipment that's used by scientists in labs, in the field, and even on the International Space Station. But a major focus is to make biotechnology more accessible for students in the classroom. Our equipment is designed with classroom use in mind, and we also offer innovative lab activities to give students real hands-on experience with biotech. miniPCR strives to create educational resources and hands-on labs that give students experience with aspects of biology that were previously inaccessible in the classroom. CRISPR-Cas is an excellent example. By now, everyone's heard of CRISPR, and it's poised to transform many aspects of human life. Because of this, it's important for students to understand what CRISPR is and what it can be used to do. Gene therapy is a great lens through which you can explore CRISPR-Cas with your students, and that's what we're going to talk about today. First, we'll cover some basic information on what gene therapy is. Then we'll dive into how CRISPR-Cas works. And finally, we'll bring those two topics together in a hands-on paper modeling activity. First up, what is gene therapy? Because genetic diseases are controlled by DNA, some genetic diseases can be treated by changing a patient's DNA. This approach is broadly referred to as gene therapy. While this is obviously a great idea, designing a successful gene therapy is pretty difficult. You need to know a lot about the genetic cause of the disease and how the disease affects the body. And you also need a way to change a person's DNA inside their cells with a high level of control. Otherwise, you could introduce unwanted mutations which could be harmful to the patient. This was a major technical limitation until quite recently, and we'll talk a lot more about this when we discuss CRISPR-Cas genome editing. To explore the power of gene therapy, we'll use the example of sickle cell disease. Remember that gene therapy can only be used to treat diseases with a genetic cause. Sickle cell disease is caused by a mutation that distorts red blood cells from their classic biconcave shape, which you can see on the left, into sickles, which you can see on the right. To understand why having sickled red blood cells is dangerous, we need to look at blood cells within blood vessels. Here you can see normal red blood cells in a blood vessel. It's really important that red blood cells can flow freely through even our tiniest blood vessels, and their flexible biconcave shape is vital for this. Sickled red blood cells are stiff and clumped together, which can block blood flow. Depending on where these blockages occur, the patient could experience extreme pain or even death, for example, if blood flow to the brain or the heart is blocked. Sickle cell disease is life-threatening, but aside from a bone marrow transplant, current treatments can only address a patient's symptoms. But because sickle cell disease is a genetic disease, it's theoretically possible to use gene therapy to change a patient's DNA and cure the disease. So why hasn't this already happened? As I briefly mentioned earlier, gene therapy can't be used for every genetic disease. A first major requirement is that scientists need to know a lot about the genetic cause of the disease and how it affects the body to be able to design a gene therapy treatment. This has actually been true for sickle cell disease for a pretty long time, but we didn't see a very effective gene therapy treatment until recently. And that's because a second major requirement for gene therapy is that scientists need a way to edit the patient's DNA in the disease-affected cells with high specificity. Anytime you're changing the DNA, there's a risk of introducing unwanted changes, and these could be harmful to the patient. Even though there were ways to edit the genome, they were pretty difficult to use in human patients. Luckily, the development of CRISPR-Cas genome editing changed the game. This brings us to the second major topic for today, CRISPR-Cas. CRISPR-Cas is used to perform genome editing, which is when scientists change the DNA sequence inside a cell. This can include adding DNA sequences, removing DNA sequences. Scientists can even change specific bases, like swapping an A for a G. And because DNA contains the instructions for life, changing an organism's DNA can change the organism. Because of this, scientists have long sought to edit the genome. As we already discussed, one application of genome editing is gene therapy, where scientists try to treat a genetic disease by changing the patient's DNA. And while scientists have been pursuing gene therapy for decades, there hasn't been much progress until recently. But luckily, between 2012 and 2013, work from several scientists came together to demonstrate the use of a powerful new way to edit the genome, the CRISPR-Cas system. There are numerous reasons CRISPR-Cas revolutionized genome editing, but we'll focus on two. Most importantly for gene therapy in humans, the CRISPR-Cas system is highly specific, meaning it allows scientists to precisely control which DNA sequences get modified within a cell's genome. We'll talk more about what makes the CRISPR-Cas system so specific in a second, but I do want to highlight again that it's essential that any changes to the human genome be made with precision to avoid unwanted mutations, which could be harmful. Second, compared to older methods of genome editing, CRISPR-Cas is pretty easy to use. I can't overstate the importance of the application of CRISPR-Cas to the field of genome editing, and in recognition for their contributions, two scientists that pioneered the technique were awarded the 2020 Nobel Prize in Chemistry. In its most basic form, using the CRISPR-Cas system for genome editing requires two major components. First, you need a Cas protein. Cas proteins are enzymes that cut DNA like molecular scissors, and while there are several different Cas proteins, scientists typically use one called Cas9 for genome editing. Cas proteins are nucleases, which is just a term for enzymes that cut nucleic acids like DNA. There are lots of different types of nucleases, but the thing that makes Cas nucleases special is that they can be programmed to cut any DNA sequence. That's because a small RNA molecule that binds to Cas9 determines which DNA sequences Cas9 will cut. The RNA molecule basically programs the enzyme. This is the second thing you need. The small RNA is called the guide RNA because it acts like a guide and tells Cas9 where to cut the DNA. It's really important to understand that CRISPR-Cas gives scientists a simple way to target a precise DNA sequence for modification. And to understand how the guide RNA specifies what DNA sequences Cas9 will cut, we need to add a little more detail to our diagram. At one end of the guide RNA is a sequence of approximately 20 bases that determines where Cas9 will cut the DNA. When this 20 base sequence binds with a complementary DNA sequence, then Cas9 will cut the DNA. Here's the same guide RNA interacting with a different DNA sequence. Because the guide RNA is not complementary to the DNA here, Cas9 will not cut the DNA. One of the biggest advantages of the CRISPR-Cas system is that it's highly specific. And that's because for Cas9 to cut, 20 bases of the guide RNA need to match the DNA. And while that might not seem like a lot, the chance that any random 20-base sequence occurs is around 1 in a trillion. And to put that into context, the human genome is only 3.2 billion base pairs long. To visualize the whole process, let's look at an animation. We'll look at a simplified schematic where the DNA is untwisted, just because it makes it easier to see what's happening. First, the guide RNA binds to Cas9. Cas9 unzips the DNA, allowing the guide access, and if the guide matches the DNA, Cas9 will cut both strands of the DNA. This is called a double-strand break. You're probably thinking, wait, I thought we were talking about editing the genome. What in the world does cutting DNA at a specific location have to do with that? To understand why creating a double-strand break at a specific location in the genome is often a first step in genome editing, we need to talk about what happens in cells after a double-strand break occurs. Double-strand breaks are really bad, and the cell has to repair the break to survive. There are two different ways a double-strand break can be fixed. The first option leads to precise repair of the DNA. The downside is that the repair process is more complex and can only happen when the cell is dividing. The second repair option is error-prone, but this error-prone repair pathway is simpler and can happen anytime. Let's use an analogy to introduce the two repair options. Imagine you have a string of colored lights and the cord gets cut in the middle. If you want the string of lights to work, you're going to need to reattach the wire. The first step would be to trim back the wire so that you have nice clean ends to splice together. And depending on how severe the damage was and where it was, this could include removing some lights from your strand. So now we have nice clean ends of wire to reattach, but we lost some lights. One thing we could do is refer to a similar strand of lights to help us with the repair. In general terms, a template is a pattern that can be used as a reference when building something. If we have another string of lights that has a similar pattern, we can line it up and know what lights are missing from where we had to cut back the wire. By comparing to the template, we know we need to add a short section with a red light and a blue light if we want our repaired strand of lights to have the same pattern. Now our damaged string of lights is repaired, and notice that it matches the template strand. One thing to keep in mind is that what matters about the template is that it matches the pattern around the break site. It is possible for a part of the template that gets copied during the repair process to differ from the original strand. In this way, repair using a template could introduce specific changes. For example, if we use this as our template, now the break is repaired with two green lights instead of the original red and blue, because in the template there were two green lights there. The other really obvious way to get the strand of lights to work again is that we could just stick the two ends back together. The pattern of lights is different now, but the lights will totally be functional. But remember, the string of colored lights is an analogy. We're really talking about how the cell can repair double-strand breaks in the DNA. The first repair pathway is called homology-directed repair. This is the precise repair using a template. A limitation of homology-directed repair is that it can only happen during certain stages of the cell cycle. But when homology-directed repair does happen, specific changes can be introduced through the use of a repair template. This gives scientists a powerful tool to introduce specific changes to the DNA. The second repair pathway is called non-homologous end joining. This is the error-prone repair where the cut ends of DNA just get stuck back together. That can happen at any time. But keep in mind that this error-prone repair pathway does introduce random changes to the DNA. Because non-homologous end joining tends to be the more common repair mechanism, and because it's easier for students to understand conceptually, we're going to focus on it in this webinar. Let's quickly take a look at what happens when a double-strand break in DNA is repaired with the just stick it back together method. The cut ends of the DNA are reattached, but random bases are usually added or removed from the DNA at the break site. Here, I'll illustrate some DNA bases being removed from the left piece of DNA. Non-homologous end joining does result in the cut ends of DNA being reattached. Remember, if the break isn't fixed, the cell will die, so it's essential that the DNA is rejoined. But I'm sure you realize that adding or deleting DNA bases are types of DNA mutation, and here we just introduced a permanent change to the genome. Often, we think of mutations as being bad, but I hope to convince you that mutations that disable genes can actually be really useful for scientists. That's because there are still many, many genes whose function remains unknown. A common method to understand the function of a gene is to disable it and then observe the effect. Scientists call this knocking out a gene. CRISPR-Cas genome editing made it possible for scientists to easily knock out any gene in any organism. And this is the most prevalent use of the CRISPR-Cas system in scientific research, and it's led to an explosion of important discoveries across all fields of biology. I'll quickly mention that miniPCR has a lab called Knockout that lets your students knock out a gene in bacteria, and I'll talk more about that at the end of the webinar. But that's not the focus for today. We are talking about using CRISPR-Cas for gene therapy. You might be surprised to find out that the gene therapies that are in use right now use knockout very widely. There are only 4 major areas currently where CRISPR-Cas has been used in recent gene therapy clinical trials. We have blood disorders, including sickle cell disease, cancer, a disease caused by a protein being misfolded, and an eye disease that leads to blindness. For these gene therapies, 3 of the 4 used a gene knockout approach. The eye disease was the only genetic disease where only precision gene editing that relied on homology-directed repair was used. There is a clinical trial using precision gene editing that relies on homology-directed repair to treat sickle cell disease, but the first clinical trial used a gene knockout approach, and I'm going to tell you all about that now. To understand how sickle cell disease can be treated using gene therapy, I need to tell you about the specific cause of the disease. The mutation that causes sickle cell disease is in a gene that has the instructions for making part of a protein called hemoglobin. Hemoglobin protein transports oxygen in red blood cells, and red blood cells are jam-packed with it. At the protein level, hemoglobin protein is actually made of 4 protein subunits, 2 alpha globin subunits and 2 beta globin subunits. The disease-causing mutation for sickle cell disease is a single base substitution in the beta globin gene. This mutation makes hemoglobin proteins associate into long strands, and that's what actually distorts the red blood cells into a sickle shape. So the goal of a gene therapy treatment for sickle cell disease would be to prevent hemoglobin protein from clumping, since that's what distorts the red blood cells into sickles and causes the problems for patients. And as I mentioned, 2 different CRISPR-Cas gene therapy approaches have already been used to treat sickle cell disease. In the first, which I will admit is more intuitive, scientists use CRISPR-Cas to cut the beta globin gene near the mutation that causes sickle cell disease. They provided an engineered template and hoped that the double-strand break would be repaired using that template DNA. And if that happened, then the disease-causing mutation would be corrected. This clinical trial is underway, but again, this is not the one we're going to focus on. That's because I want to focus on the first use of CRISPR-Cas gene therapy to treat sickle cell disease in a human patient. The first patient to receive this gene therapy, Victoria Gray, is pictured here, and there are some really amazing interviews with Victoria that I encourage you to share with your students to put this amazing medical advancement in a more human context. This clinical trial took a gene knockout approach. I'll mention another reason I think this is a really nice clinical example to cover with students is that this gene therapy actually treats 2 diseases, sickle cell disease and another disease called beta thalassemia that's caused by a different mutation in the beta globin gene. In this gene therapy strategy, scientists use CRISPR-Cas to cut a different gene, with the goal that the double-strand break would be repaired using the error-prone stick-it-back-together method, non-homologous end joining. The key to this strategy is that scientists selected a gene that, when it's knocked out, will indirectly prevent hemoglobin protein from clumping, thus treating the cause of sickle cell disease. Early results from this clinical trial have been published and are quite promising. To help students understand how CRISPR-Cas was used in this gene therapy treatment for sickle cell disease, we created a hands-on paper model activity. The paper model comes with all the background information you need to teach your students about how this gene therapy treatment works. We're going to walk through the background first, then I'll show you the paper model in action. To understand this sickle cell gene therapy treatment, I need to tell you a little bit more about hemoglobin. Interestingly, during fetal development, we produce a different form of hemoglobin called fetal hemoglobin. Fetal hemoglobin has 2 alpha globin subunits, but instead of beta globin, it has 2 gamma globin subunits. Beta globin and gamma globin are functionally quite similar. Around birth, a genetic switch flips and our cells stop making gamma globin and start making beta globin. The key to the gene therapy treatment is that the gamma globin gene is still in our cells, and if gamma globin is produced, fetal hemoglobin is entirely functional in adult cells. There are actually some people with a mutation that makes them have persistent fetal hemoglobin production throughout their lifetime, and this has been shown to protect them from sickle cell disease. The thought is that since beta globin and gamma globin compete to bind with alpha globin, the presence of some gamma globin will prevent the formation of the really long strands of mutant hemoglobin, as shown here. Scientists reasoned that if they could make adult red blood cells produce fetal hemoglobin again, that could counteract the effects of the sickle cell mutation. And to accomplish this, they disabled the genetic switch that turns off gamma globin production. Another protein called BCL11A inhibits gamma globin production. Around birth, a genetic switch flips that turns on production of BCL11A. This decreases gamma globin. So before birth, the BCL11A switch is off, which means there is no BCLA protein. And in the absence of BCL11A protein, gamma globin can be produced. But after birth, the BCL11A switch is on, which means BCL11A protein gets made. In the presence of BCL11A protein, gamma globin cannot be produced. Scientists use CRISPR-Cas to disrupt the BCL11A regulatory switch in blood cells. They hoped that this would decrease BCL11A protein, which should allow gamma globin protein production. Bear with me here because the explanation for how this works involves a double negative. Remember, the goal is to increase gamma globin. We're going to work backwards from this goal. To increase gamma globin, we can decrease the production of BCL11A protein because BCL11A is an inhibitor of gamma globin. In essence, we want to inhibit an inhibitor. To prevent BCL11A protein production, scientists set out to break the BCL11A regulatory switch. So, how did they actually use CRISPR-Cas to break this DNA switch in the cells of sickle cell patients? To understand how scientists use CRISPR-Cas to edit the genome of sickle cell patients, there's one more thing you need to know. And that is that the stem cells that give rise to our blood cells live in our bone marrow. Because red blood cells cause the symptoms of sickle cell disease, if you could edit the DNA in a patient's blood stem cells, then those stem cells should go on to produce normal red blood cells. In essence, a patient can serve as their own bone marrow donor. This has the huge benefit that there aren't concerns of immune rejection. And this is the basic workflow for gene therapies that treat blood disorders, including sickle cell disease. First, you collect a patient's blood stem cells, and then you modify the cells using CRISPR-Cas outside of the body. Modifying the cells outside of the body gives scientists a chance to check them and make sure that the genome editing had the intended effect before the cells are put back into the patient. Once scientists have confirmed that they have successfully edited the genome of the blood stem cells as intended, drugs are used to kill all of the patient's remaining blood stem cells. and then the gene therapy treated stem cells are reintroduced into the body. If the gene therapy works as intended, now the patient should make healthy red blood cells and be cured. This step 2 is the step that we're going to model in the paper model, where scientists use CRISPR-Cas to edit the DNA in a patient's blood stem cells. So now that we have this background information down, let's actually see the paper model in action. The paper model has 3 basic parts: the Cas9 protein, the guide RNA, and a strip of DNA. First, we'll focus on the Cas9 protein. This serves as the base of the paper model. Remember, the first thing that happens during CRISPR-Cas genome editing is that the guide RNA binds to the Cas9 protein. Now let's focus on the cutout of the guide RNA. The 20 bases that will guide Cas9 to cut the BCL11A regulatory DNA in the right place are shown here. And I will note that this is the actual sequence that was used in the sickle cell gene therapy treatment. We're going to place our guide RNA onto the Cas9 protein in this yellow region that matches the size and shape of the guide RNA cutout, and then just tape it down so that it stays in place. Recall that once the Cas9 guide RNA complex forms, Cas9 scans the DNA until it encounters a DNA sequence that's complementary to those 20 bases in the guide RNA, and then Cas9 will cut the DNA. Now let's take a look at the DNA strip. We have a very small region of the BCL11A regulatory DNA. And there are 2 things that I want to highlight on this DNA strip. The first is that in the middle of the DNA strip is a short region where the sequence is bolded. Previous research had identified this as a kind of Achilles heel region for the BCL11A regulatory DNA. Scientists had already shown that when mutations were introduced in this specific region, it broke the regulatory switch and BCLA11 protein was no longer made. And remember, this is the goal of this gene therapy treatment. We want to prevent the production of BCL11A protein because BCL11A protein prevents the production of gamma globin. And our therapeutic goal is to turn on gamma globin production so fetal hemoglobin is present again in adult red blood cells. The second thing I want to highlight is the presence of these purple boxes. There's one last thing I didn't tell you about how Cas9 works, which is that it actually doesn't check every single position in the DNA looking for a sequence that's complementary to the guide RNA. It only checks DNA that is adjacent to something called the PAM sequence. The PAM for Cas9 is NGG, where N can be any base. All the PAMs are marked on the DNA here with purple boxes, and you can see that there are a lot of PAMs and they're on both strands of the DNA. For example, here's a PAM that's AGG, and then the next one we have is TGG. The fact that there are so many PAMs is actually really useful for scientists, because when designing a guide RNA, it needs to be adjacent to a PAM. But as you can see, even in a short piece of DNA like this, there are a lot of PAMs. So there are typically many possible guide RNAs that could work to target even a short piece of DNA. Now we're going to slide the DNA under the guide RNA so that we can still see the sequence on both strands of DNA. To model how the PAM works, we just line up the purple PAM box that's on the Cas9 protein with a purple PAM box on the DNA. And then we would check to see if the guide RNA is complementary to the DNA in that position. This first PAM site isn't the one we're looking for because the DNA sequence in the cutout doesn't even extend this far. So we'll just move down to the next PAM. Here, the first pairing is a C with a T, so that's not a match. We would just slide the DNA down to the next PAM and check again. but that was actually the last PAM on this strand of DNA. So we didn't find a complementary place for the guide RNA. I'm just going to flip the DNA over and now we'll check the other strand. Using this first PAM, I can already see that I have a match. When comparing the gRNA with the bottom strand of the DNA, I have a C pairing with a G, a U pairing with an A, and so on. In fact, the entire length of the guide RNA is complementary with the DNA sequence here. When you find the place where the guide RNA is complementary to the DNA, these arrows on the Cas9 protein indicate where Cas9 will cut the DNA. I'm just going to mark it, take the DNA out, and then cut it with scissors. So let's take a step back and think about what's happened so far. Remember, the therapeutic goal was to use CRISPR-Cas to disable the BCL11A regulatory DNA in the Achilles heel region. To accomplish this, scientists designed a guide RNA that would tell Cas9 to cut the DNA in that Achilles heel region. That's where we're at so far. Right now, we've introduced a double strand break. And remember, these are bad news. The cell needs to repair this break or it will die. And recall that the most likely thing that will happen is that the cut ends of the DNA will get stuck back together with the error-prone non-homologous end joining repair method. To model non-homologous end joining, students are given the choice to trim off up to 10 DNA bases on either side of the double-strand break. Remember, non-homologous end joining introduces random mutations, insertions or deletions around the cut site, and so there's no right or wrong answer here. I feel like cutting off this many bases just from this side of the DNA. What I'm doing is introducing a random deletion. To finish modeling non-homologous end joining, all I need to do is tape the cut ends of the DNA back together. Now the repair is complete. The BCL11A regulatory DNA is reattached, but a random mutation has been introduced in the Achilles heel region, which was our therapeutic goal. Now, if these edited cells are put back into a human patient, the BCL11A regulatory switch is broken, so the BCL11A protein will not be produced in red blood cells. In the absence of this inhibitory BCL11A protein, the red blood cells will produce gamma globin protein, and fetal hemoglobin will be present to counteract the effects of the sickle cell mutation. The paper model also comes with a blank guide RNA that students can use to design another guide RNA that could have been used to accomplish this same goal. I'll switch out the guide RNAs now and put in a new DNA strip. All you need to do to design a guide RNA that has the potential to treat sickle cell disease is to pick a PAM that places the cut site within the Achilles heel region of the BCL11A regulatory DNA. You can use either strand of DNA. I'm just going to randomly pick this PAM. Once you line things up and make sure that this PAM would lead to a cut in the Achilles heel region, all you need to do is use the simple base pairing rules to fill in the guide RNA sequence. I have an A here on the DNA, so I need a U on the guide RNA. Remember, RNA uses U instead of T. Another A on the DNA, another U for the guide RNA, and I just continue along and fill in the bases that are complementary. G, C, U, U, and so on, until I have the whole 20 bases. And just like that, your students can see how easy it is to design a guide RNA that will tell Cas9 to cut a specific DNA sequence. You basically just need to understand the simple base pairing rules. So one of the most exciting things about CRISPR-Cas genome editing is how it's been used to make major breakthroughs in both basic and applied science. And we actually made a paper model for each. We just walked through the example of using CRISPR-Cas gene therapy to treat sickle cell disease. And to dive into a fun example of basic science research, we have an activity where students model research on snails. In this activity, students explore how scientists use CRISPR-Cas to knock out a gene in snails to understand the genetic control of whether snail shells coil to the left or to the right. As a former researcher, I love exposing students to examples of basic scientific research like this. Plus, this activity has the benefit that the background information is not as complex as with the sickle cell gene therapy. Both of these paper models are totally free. You can download them off our website, and they're designed to work as standalone activities. So each paper model contains all the necessary background information for your students to understand the activity. Another great free resource from miniPCR is the DNA Dots series. DNA Dots are 2-page explanations of molecular biology topics that are written using non-technical language. They make great ready-to-go assignments because they come with review, critical thinking, and discussion questions. And there's a whole library of DNA Dots available, including a great one on CRISPR-Cas. miniPCR also has lots of free educational webinars. A topic that could be great to cover in an advanced class is the natural function of the CRISPR-Cas system and the path that led students to apply it to genome editing. I encourage you to check out my colleague Emily's free webinar, which covers both of these topics, and it even comes with a free student worksheet. On a final note, if you really want to take things to the next level and perform a CRISPR-Cas lab, miniPCR does have 2 options. In the knockout lab, students use CRISPR-Cas to knock out a gene in bacteria and observe the resulting phenotypic change. And in the chopped lab, students explore how guide RNAs are used to program Cas9 to cut specific DNA sequences in an easy test tube reaction. We have separate webinars that describe each lab in great detail. And that's it. Thanks so much for joining me. The QR code in the corner will take you to the miniPCR CRISPR-Cas resources landing page, where you can access all of the free CRISPR-Cas resources from miniPCR, including the paper models.